Thank you, Eugenie, for the very nice introduction, and thank you all for inviting me today to do this presentation. My name is Pen, and I'm a postdoc at Fred Hutchinson Cancer Center working with Professor Jingyi Jessica Lee. Today, I would like to introduce a recent work of ours aimed at enhancing the rigor of metacell partitioning in single-cell data analysis. Our work is called mcRigor, where MC stands for metacell. Well, let's start with a quick recap. For people who are not so familiar with single cell and the metacell concept, here is like a brief introduction. Here, this top left panel shows the typical format of a single cell RNA sequencing data, which is basically a matrix of gene expression values with genes shown as rows here and cells as columns. We know that in this data matrix, there could be like many zeros, like around 90%. And many of them are due to the imperfection of single cell sequencing technology, which means that for many genes that are expressed in the cells, because of the low per cell sensitivity, these genes expression are not captured. So these are shown as zeros or as the white blank blocks right here. So intuitively, we do not want these zeros in our downstream analysis, or to say we want to solve this kind of sparsity issue, because this can hinder many downstream analysis tasks. And this metacell concept we're talking about today is exactly a pre-processing step in single-cell data analysis pipeline that is developed to solve this kind of issue, this sparsity issue. So what metacell does is that it tries to identify similar single cells and shown as the colored dots right here within one black circle on the bottom right. And then we just aggregate these similar cells into a metacell by averaging. So here on this bottom right panel, each black circle you can think of as a very small cluster represent one metacell. Then we just need to define the average profile for each black circle, and we will end up getting this gene by metacell matrix instead of the original gene by cell matrix. And by doing so, we usually can reduce the sparsity level from like around 90% to only like 50%. But at the same time, we also pay the price, which is now we have a much smaller number of observations. We have metacells instead of single cells, so the resolution is lower. But there are also cases where people think having these like fewer observations or analysis units is not always a bad thing, because this would usually mean that we can have better scalability. So, or to say we have lower computational cost. So, well, due to all these reasons, the metacell concept is quite popular in single-cell data analysis and has been used in many studies, as I will show in short. But one question that motivated our research is what is a reasonable definition for aggregating single cells into metacells? Because if you think of like two extremes, one is that if we just aggregate all the cells into one metacell, then that won't make any sense. Because we just totally lost the single cell information. Or on the other extreme, if we just leave the single cells as they are, we're just not doing any kind of aggregations. And there must be some sweet spot in the middle. So we wish to answer this question, like how can we find this sweet spot? And this slide lists the existing use of metacell idea. There are 5 general methods metacell methods so far, which have been developed into the software package that people can just directly use. Starting from the very first version, the metacell, and the second version, metacell 2, by the same group. And there are also supercell, the C cells published last year, and the most recent metaQ published this. C cells was published in 2024 and 2023, and this metaQ was published last year. And besides these 5 general methods, we also found that metacells have been used in many in-house data analysis published in high-impact journals like Nature. They have either come up with some very straightforward ways of defining metacells or just use one of these 5 general methods. But looking at all these papers, we do not really see a consensus on metacell definition. People can just do it in their own way. So motivated by this background, we developed our mcRigor method because we wish to improve the rigor of metacell definition. Generally, we wish to answer three questions. First is how to define a metacell. Second is how to find dubious metacells generated by existing methods that do not follow the appropriate definition. And third, we want to answer how do we optimize metacell partitioning for a given dataset. And just in short, our method is designed as a way to help with existing metacell methods to gain better metacell partitioning results, but it is not a completely new algorithm to build metacells from scratch. So it is more like an add-on concept to make existing methods more rigorous. Okay, to answer the first question, how to define a metacell, we go back to the very first paper that proposed this metacell concept. There it says a metacell should be defined as a homogeneous collection of single-cell profiles that could have been resampled from the same original cell. Well, this is a biological definition, and we wish to first translate it into something statistical. So our translation is that given this concept, if we see variations of single cell within one metacell, then these variations should be exclusively due to measurement error, or to say technical error, instead of biological differences. So given this definition, how do we address the problem? Well, I think the question now becomes how to actually define the measurement error in single cell sequencing data. We actually use this paper's formulation, which is a very nicely written paper about single-cell statistical modeling. There, the authors separated the single-cell data into two layers. First is an expression model, and the second is a measurement model. And they two together give us a model that we can get our observed data. Or more specifically, an expression model describes the features' true expression levels in cells, while the measurement model is literally like a conditional distribution. That is like given the cell's true expression, what are the observed count we can get in our data. And we formulate this two-layer model into statistical language. Here we consider N cells as observations and P genes as features. The expression model is about the true gene expression in cell I represented by lambda I, where lambda I is the p-dimensional vector, each dimension for one gene, because we usually look at the p genes jointly, in this expression layer. And the expression of these p genes are assumed to jointly follow from some distribution about the biological states of the cells, represented by xj here. I also want to point out that the lambda represent the relative expression level with this component summing up to 1. We focus on this relative expression instead of the absolute expression because we believe a single cell sequencing can only provide meaningful expression about the relative abundance instead of the absolute abundance. Then given this lambda I, the measurement model depicts the variation of or errors introduced in the sequencing procedure. Like given the true expression, lambda, how do we end up getting the observed counts, Yij? And this is typically assumed to follow this Poisson distribution. On the right-hand side, the Ci represents the expectation of cell library size Yi plus, also called per-cell sequencing depth. So based on this two-layer model, our statistical definition of a metacell is a group of single cells that share the same lambda. So the cells in the same metacell have the same lambda, but they have different yi's. Then we can formulate this into a statistical question. Can we check if one existing metacell actually satisfies this definition? If yes, we will call this metacell trustworthy, and otherwise we will call this metacell dubious. And just some more motivation of why we want to identify dubious metacells. This is again a figure from the review paper that we used in introducing single cell and metacell concept. Here, the single cells are colored by their cell states. Each black circle represents one metacell, and you can see that this middle one is one mixed by cells from two different states, and it is clearly a dubious metacell. So here, if in our analysis, if we're interested in studying the association between gene A and gene B, which is a very common and important analysis for single cell data analysis, and you can see that based on the single cell level data on the top right, there is in fact no association between this gene A and gene B, at least no positive for sure. But if we compute the association at metacell level with one dubious metacell, then this dubious one will lie here in the middle and we will get this fake positive association. This is just an example of the kind of dubious that can be caused by dubious metacells, the kind of bias that can be caused by dubious metacells. And that is exactly why we want to identify these dubious metacells to avoid these bias being carried down to downstream analysis. So we propose our mcRigor method, which generally gives a statistical criterion that can identify dubious metacells consisting of cells from different states. And after identifying these dubious metacells, the next step could be to optimize the hyperparameter of existing metacell methods. We focus on the granularity level hyperparameter called gamma here, because this is the most important hyperparameter that is used by all metacell methods. It is defined as the average number of single cells within one metacell, or like the metacell size on average. We hope to choose this gamma in a data specific way. At the most basic level, we use feature correlation matrix as a way to build our criterion. The intuition is that if we look at the cells that constitute a trustworthy metacell, its feature correlation matrix will be nearly diagonal, like the one on the bottom left. But for a dubious metacell, there could be a clear block structure within this correlation matrix. So how much this feature correlation matrix deviates from its identity matrix could be a guide to help us identify the dubious metacells. Well, more specifically, we would first need a per metacell statistics that can depict how much likely is this cell dubious. Well, based on the feature correlation matrix that I just mentioned, and we call the statistics the MCDIV, standing for metacell divergence. Then we also need to construct a null for the statistics, a null that would mimic the trustworthy metacells, so that we can use it to evaluate the significance of our MCDIV statistics, or how significant is each metacell dubious. We use a permutation-based strategy to do so. And another important point here is that in the null construction, we want to preserve the cell library size. And the reason for this is that the cell library size is involved in this measurement error as the sum total of the multinomial distribution. And we know that the sum total would induce a small level of feature correlation that we need to account for. Well, in more details, we call this null construction approach the double permutation approach. This is because simply permuting within each gene won't be enough in our case because it would break the cell library sizes, which are the row sums here. So instead, what we do is we permute within each cell to preserve these cell library sizes while still removing the feature correlations. However, since this kind of permutation alone would make the null statistics not really comparable to original ones, as these new columns would become permuted genes instead of the original ones. So to make them comparable, we actually add a second layer of permutation, which is called the within gene permutation as the normalization step. Then the top row and the bottom row could form a comparable pair. That is, we obtain our statistics MCDIV from the top row and the corresponding MCDIV, the corresponding null statistics MCDIV null from the bottom row. Right, so here is exactly how we compute these MCDIV and MCDIV nulls. So specifically, we compute a pair of MCDIV and MCDIV null for each metacell. And here we use two metacells for illustration. One is this purple trustworthy metacell, and the other one is this green dubious metacell. Here, this rectangle in this top left box represent the matrix, the gene expression matrix from this trustworthy purple metacell. Here with genes as columns and cells as rows. And the square matrix in gray colors represent the gene-gene correlation calculated from the corresponding data expression matrix. So our MCDIV statistics is computed as the Frobenius norm of the difference between the original feature correlation matrix and the identity matrix. And we divide it by the Frobenius norm obtained after within gene permutation to normalize it, as we just mentioned. So the MCDIV is this part divided by this part. And for MCDIV null, we just do the same thing, but this time on the data matrix after within cell permutation. So the MCDIV null is this part divided by this part. Here you can roughly tell that for the trustworthy metacell, the values of MCDIV and MCDIV null would be similar, but for the dubious metacell, this MCDIV value would be much larger than its corresponding MCDIV null value. And then this figure shows you a clearer big picture of our method. We do the computation for each metacell from metacell 1 to metacell m. Then by pulling all the MCDIV null values all together, we can derive a threshold for the MCDIV null statistics, shown by this red curve line. So it's like if the MCDIV of one metacell passes go above this threshold, we would consider it as dubious metacells. And if it goes just below this threshold, we consider it as a trustworthy metacell. And we also noticed that this MCDIV null derived threshold would depend on the metacell size. That is, our threshold is determined as the 95th quantile of MCDIV null conditional on the metacell size m. So this is the pipeline of the first functionality of our mcRigor method, which is to identify the dubious metacells. And here is one interesting application of our mcRigor on this data set of blood cell gene expression from both healthy people and COVID-19 patients. This data was originally used to study the gene-gene co-expression differences between the two cohorts. Since the metacell are believed to be able to strengthen the co-expression signal, we applied a supercell metacell method to this data and compute the gene co-expression over the generated metacells. Something we want to pay attention to here is this gene module in this yellow box, defined as the adaptive immune response module. First, we can compare the single-cell level co-expression on the left with this metacell level co-expression on the right, and we can see that the supercell indeed strengthens the correlation signal in both healthy and COVID-19 patients. But there is no much differences in this adaptive immune response module between the two cohorts. However, if we use our MCDIV method to remove the dubious metacells, which give us the co-expression profile in the middle, then we can see that there is a clear difference between the two groups, which implies that the gene in this module would tend to co-express more strongly when the patients are infected. And this is consistent with the fact that these genes are adaptive immune response genes. Then going back to the data, we found that the reason why there is no significant enrichment when all metacells are used is that these dubious metacells, shown as this pink spots, they falsely drove the correlation to be really high in the healthy group. And our mcRigor is able to remove them to eliminate this kind of bias and eventually being able to detect this module gene co-expression enrichment. And here's another application about association analysis between chromatin accessibility and gene expression. Well, this analysis is mostly used to identify the enhancers of genes. Here we use the SC multiome data, which include paired SC-ATAC-seq data and SC-RNA-seq data. And in the top left panel, these peaks represent the chromatin accessibility signals obtained from the single-cell ATAC-seq data. Here we just focused on the chromatin region near the GATA2 gene. And the black and the purple curves in the bottom left panel represent the significant associations found between peak accessibility and the GATA2 gene expression. And this analysis was originally performed in a recent C-cells paper, where they show that at a single cell level, in this row, we can actually we usually cannot identify that much enhancers or these purple curves. But if they use the C-cells method to aggregate cells, single cells into metacells, we can usually identify much more enhancers. However, what we found is that if we go further to apply our mcRigor method to remove the dubious metacells, we can actually further rectify the results. Specifically, we are able to remove a peak, this red one called peak 2, that has very low signals across all cell types. And if we look at the original single cell data, the non-zero chromatin accessibility of this peak of this GATA2 gene marked in this red, in this yellow circle, actually shows non-significant correlation. And these evidence support us in saying that maybe this peak indeed should not be identified in the very first place, and our mcRigor did a good job in removing it. And also using our mcRigor, we are able to identify one additional highly correlated peaks with the GATA2 expression, which is this blue one. And this peak indeed shows some significant correlation with the GATA2 expression at single cell level. And actually this peak has already been validated as an enhancer for GATA2 in the previous studies. And all of these results affirm us that our mcRigor methods can further rectify the results to make the whole analysis more rigorous. And so far we have talked about our mcRigor's first functionality, how to identify dubious metacells. Now I wish to introduce how to achieve the second goal, which is how we can nominate the top performing metacell methods and at the same time optimize its hyperparameter, the granularity level. And the main idea is that for a given data set, we would compute a score for each metacell partition generated by a specific method under a specific hyperparameter setting. The method and hyperparameter setting that receives the highest score would be considered as the optimal one for the given data set, here marked with this red dashed line. Then the question is like how we can define the score. Well, previously we mentioned that bigger metacells or larger granularity level can usually better resolve the sparsity, but at the same time it may distort the data. And since there will be more dubious metacells. And on the opposite side, a smaller granularity level can leave the sparsity issue unsolved. But the smaller metacells usually comes with like less dubious metacells, so less signal distortion. So this is saying like choosing the optimal setting is usually just essentially about doing a trade-off between the sparsity and signal distortion. So here we use this zero rate or it is actually just a proportion of zeros to assess the sparsity level left in this metacell profile. And we use this dub rate, which is the proportion of single cells that constitute dubious metacells, to assess the level of signal distortion. Then we just use 1 minus the sum of these two scores as our evaluation score to choose the best method and hyperparameter that receives the highest score. And here on the right, we have proved using our simulation data that mcRigor can accurately find the optimal hyperparameter that best align with the ground truth. Well, this is just one example to show you how our mcRigor can help optimize the hyperparameter in the D gene detection analysis tasks. Well, this D gene detection is actually one of the most important tasks in single-cell data analysis. It mainly aims to find genes that have the different expression levels between two cell types or two different conditions, so that these genes could be of more interest in further analysis. Here we use the single-cell RNA sequencing data with paired bulk RNA-seq data. We know that since the bulk data are obtained from a lower throughput technology of less measurement error, we usually believe the DE genes detected from the bulk data is more reliable. So here we just use bulk data as a gold standard to evaluate our DE gene detection given by the metacells generated from different methods under different gamma values. We identify the DE genes between two cell types, while represented by this we just identify DE genes between two cell types, and here we compute the F scores of DE genes resulted from different partitions versus the DE genes derived from the bulk RNA sequencing data. And you can see that the best one is the one with gamma equal to 13 and marked by this this red dash line. So this is the one given by the purple color, which represent the C cells method. And going back to our mcRigor results on the top, in the top panel, we can see that the C cells with gamma equal to 13 is exactly the optimal method hyperparameter setting that was chosen by our mcRigor method. And also, if we visualize this expression of the top bulk DE genes under different metacell partitions, like in this heat map, with rows as genes and columns as cells, we can also see that the one given by C cells with gamma equal to 13 would give us the expression pattern most similar to the bulk pattern. So this evidence shows us that our mcRigor was able to select the best metacell method and have a parameter for this data set. So just to summarize my talk, we have answered 3 questions raised in the very beginning. Our work was able to give a statistical definition of metacells based on 2-layer statistical model, and we're able to detect dubious metacells using pre-metacell MCDIV statistics and a null constructed using the double permutation strategy. And finally, we are also able to optimize our metacell partitioning by balancing sparsity and dubiousness in each data set. And our work has been published on Nature Communications last year. And there are more analysis results in our paper. You're very welcome to check them out. So with that, I'd like to thank our grant sponsor and also the members of our group for supporting this work. And I'd like to thank all of you very much for your attention.